Welcome back. We are starting with chapter 8 and we're going to discuss about the control of microorganisms in the environment. So, in this chapter, in the first part, we are going to talk about the microbial growth, replication, and the targets of control, have a generalized idea, and then we will go into the detail about some of these compounds which are essentially required for killing or inhibit the different types of microorganism. So in this big chart, what we have that these are the common microbial control methods. And before going into the detail or the examples of that, let's review some of these definitions. And these definitions are actually found at the very bottom of each of the process, and I will just explain that what I mean by that. So the first thing is that the disinfection is defined as the destruction or removal of vegetative pathogens. That means these are actively dividing pathogens, but not bacterial endospores, which are in the dormant form, usually used only on inanimate objects as well. So you cannot use it on your skin or internally you cannot use it. So this is the process of disinfection. And which are the processes? Here you can see that in the white, this is disinfection, disinfection, disinfection. These are some of the processes and we're going to discuss those in detail. The next is the sterilization. These are the processes which can sterilize, all in this black background with white lettering. Sterilization is the complete removal or destruction of all viable microorganisms. And this is also used on inanimate objects. So the difference in between that, see that in the disinfection, you killed all vegetative pathogens, but not the bacterial endospore. Sterilization will destroy all the vegetative pathogens, of course, along with all bacterial endospores. Then we have another definition of antisepsis. Antisepsis are the chemicals which are applied on the body surface to destroy or inhibit vegetative pathogens, but they are not administered internally. It is only on the body surface. So you can see that right here is the antisepsis. Right here, these are some of the antisepsis. And the agents which are used in this antisepsis process are known as the antiseptics. The next one is a chemotherapy, which is in this yellow highlighted area. We are not going to discuss that in this chapter. That will be the topic of the next chapter. So when we think about the chemotherapy, the first thing it comes to us is that it is usually used very commonly in cancer treatment. We always refer to that chemotherapy is related to the cancer, but it is not. It is much more generalized than that. The definition is the chemical used internally to kill or inhibit growth of microorganism within the host tissue. So all your antibacterial, antiviral, antiprotozoal, antifungal, all of the medication, and all of these are prescription medication that you take orally, internally, or injected to you. These are the chemotherapeutic agents, and in broadly we divide them depending upon on which they work, and we're going to talk about that in the next chapter. But we will mainly talking about the other processes which we do not take internally. So let's first take a look at that what are the different four categories of microbial control. So here what we are looking at, the first one right here are the physical agents. And the physical agents, the first area we are looking at is the heat. So the heat could be a dry heat, which could be incineration or burning. That will cause sterilization. Or dry oven or dry heat, that can cause also sterilization, destruction of everything, including endospores. Then you have a moist heat, and this is steam under the pressure, which can cause sterilization as well. We're going to talk about that. And then you have another moist heat, which could be boiling water, hot water, pasteurization. This can lead to disinfection. That means it can kill the vegetative pathogens, but it will not sterilize it. So this is the scheme for the heat. Next physical agent other than the heat is the radiation, and the radiation can be two types. One is ionizing radiation. Why we call as ionizing radiation? Because when they hit some of these microorganisms, they will knock off electrons from their atoms, what their constituents are. And these ionizing radiation split the atoms into the ions, and that's why they're called ionizing radiation. Examples are X-rays, the cathode, or the gamma rays, beta rays, and the gamma rays. These are the ionizing radiation, and these can cause sterilization as well. On the other hand, the non-ionizing radiation do not cause this type of knocking out of electrons from the atoms, but instead there are some damage to the DNA, and ultraviolet is responsible for that. That can cause disinfection, but not, again, sterilization. So we're going to discuss all of these in detail. The next group is the chemical agent. So physical agent, heat and radiation. Chemical agent, first we are looking at the different types of gases. There are some gases which are used which can completely sterilize, and some of the other gases are used for disinfection as well, but not sterilization. On the other hand, the liquid, if we use the liquid on animate object, one group, again, this could be liquid or solid as well, of course, chemotherapeutic agents, and these are your different drugs that we take, which could be liquid or solid, that we are not going to discuss. On the other hand, if we are using them on the surface, these are your antiseptic agent, and the process is known as the antisepsis. That is this process, destroy or inhibit vegetative pathogen. On the other hand, the liquid, if we just use on inanimate object, some of these liquids can disinfect. Think about just a common household spray that can disinfect surface. Also, there are some chemicals which can lead to sterilization as well. So these are your chemical agents, and we'll go detail of each one of them in a minute. The next one is a mechanical removal of the pathogens and filtration. So the filtration could be two types. One is air or particulate filtration that can lead to sterilization as well. But here I would like to say that there are some other methods of filtration where disinfection is the purpose that we use, and not always sterilization. Because if you just think about a mask that you are wearing, or you have an air handling system in your house or hospital, these can actually disinfect or reduce the amount of pathogen, but not necessarily sterilize. So this is a little bit of addition. Just remember that it is not 100% that how it is. Not all air filters can lead to sterilization. There is a good number of air filters which are disinfecting air filters, reduce the number of pathogens, but cannot sterilize. On the other hand, for the liquids, the same exists. There are many types of filtration which cannot sterilize, but there are some which can absolutely cause sterilization. Now here you have to understand one important thing, that the filtration can take care of all eukaryotic pathogens, including bacteria. But there are just one group of bacteria, which is called Mycoplasma, doesn't have peptidoglycan on their cell wall, and they do have a pliable structure, cannot be sterilized by the filtration, and virus-free liquid cannot be produced through this sterilization. So except virus and the Mycoplasma, which is a group of bacteria, all other can be sterilized. So this is an important consideration, that although we are talking about sterilization, but it is not 100% that we cannot get devoid of all different types of microorganisms as well. But on the other hand, if you just look at these dry, moist, heat, or the different chemicals, that will get rid of all different possible microorganisms, including these Mycoplasma viruses that we just talked about. And another new area, which is a biological control, is coming into the picture lately, but it is not widely used. So one of them is a predator microorganisms can be used as an antisepsis. Viruses can be used as an antiseptic agent, but again, the usage of these are very limited, not approved for usage on the human being in the United States, and some of them are used on the food products, but not on the human beings. But some of the countries do use these. On the other hand, there are some toxins which are known as a bacteriocins produced by some of these pathogens. Those can be used to completely sterilize some of these or kill all different types of life forms. But again, these are some of the very new developing area that we are going to talk about at the very end of this chapter. So now let's take a look at some of the definitions of frequently used terms. We already talked about the sterilization as well as disinfection. And then we are introducing another new term, which is sanitization. So the sanitization is a very common word that we use in the restaurant industry or the food industry. This is the reduction of microbial population to levels deemed safe. This is based on public health standard. So for example, when we do this dishwashing in a public restaurant, say for an example, we do this process to reduce the levels of pathogens to public health standards. That does not mean that the sanitization process removes 100% of the vegetative cells. That's the difference between the disinfection and the sanitization. That sanitization reduces the microbial population to a level which is safe. On the other hand, we already talked about the antisepsis, prevent the infection of living tissue by the microorganisms, and antiseptics are the chemical agents that kill or inhibit the growth of microorganisms when applied to the tissue. Most of the time, the antiseptics are used on the skin surface though. So here what we are looking at the impact of the biocide exposure, and the shape of each curve reflects the various conditions that influence the biocide effectiveness. In each case, the eventual decline in viable microorganism can occur as a stage decline of viability from antisepsis to sterilization. So if you look at that over the period of time, this scale, the Y scale, is the log scale of the microbial population, and the decrease in microbial population is along the Y axis over the period of time. So you can see that all the three lines start from the same level of microbial population on a log scale. The first line actually drops rapidly in a straight line and then levels off at the sterilization level. The second line is a sigmoidal curve gently sloping through the antisepsis layer and then through the sanitization layer, and then gently sloping and leveling off at the disinfection layer. And the third line you can easily see is a gentle arch which goes through the antisepsis, sanitization, disinfection into the level of sterilization. So this basically gives you an effect of the biocide exposure to the different types of microorganisms. So then we are looking at the chemotherapy, use of chemicals to kill immediate growth of microorganisms within the host tissue that we already talked about. So now we are going to talk about some of these two suffixes. So a cidal agent actually kill the microorganism. So these are usually used with a suffix "cide", indicates the agents that kill. So for an example, bactericide will kill bacteria, fungicide will kill the fungus, virucide will kill the virus. There is another term which is also commonly used, is known as a sporicide, which is going to kill the spores. So on the other hand, the static agents inhibit growth. So the static suffix indicates the growth inhibiting agent. Say for example, bacteriostatic or fungistatic or virustatic. So what we mean by these, that when that static agent is removed from the vicinity of these microorganisms, they do have the capability of growth. So the difference between these two are the cidal agents kill, whereas the static agents inhibit or stops growth. So let's take a look at some of these pattern of microbial death, also a pattern of microbial growth. So it is a mirror as such. So if we look at that, the microorganisms are not killed instantly, and the population death usually occurs exponentially. Why this is, remember that when we talked about the growth of microorganism, you already saw that after the initial lag phase, the growth happens exponentially in a log phase. If there is availability of nutrients, growth occurs in a logarithmic scale or exponentially. The same way that when the microbes are killed due to exposure to some agents, they are also killed exponentially, but they are not killed instantly as well. So they do have a lag phase, just like their growth as well. So we're going to use some specific terms and going to define what they are. So the measure of the agent or antimicrobial agent's killing efficiency is determined by a decimal reduction time, which is a time to kill 90% of the population of the microbes. So we're going to see that. And must be sure that they are viable but non-culturable cells are dead. So this is very important. Once they recover, they may regain the ability to reproduce and cause infection. So that's the important part. Let's take a look at how we measure the decimal reduction time, or the d-value. So for an example, in a semi-logarithmic plot, there the number of microorganisms that survive heat exposure is on a log scale on the y-axis, and the x-axis is not logarithmic and is plotted against time exposure to the heat. So the d-value is the time required for the line to drop by 1 log scale and 1 log cycle, or tenfold. So the plot on the left-hand side shows that this is an exponential plot of the survivor right here versus the exposure to heating at 121 degrees Celsius. So this is a straight line of 10 to the power 6 microorganisms at 0 minute to -1 microorganisms at about 7 minutes. So in this example, the d-value is 1 minute. The data is tabulated right here. So the d-value is the time required to reduce the number of these population by 1 log value. So that's your d-value. So it is also possible to determine the temperature change at a given d-value that decreases the microbial population by 1 log cycle. So this plot is an exponential plot of the d-values versus the temperature. So here in the y-axis, what you have is the log 10 of the d-value, and here is the temperature change at a given t-value. The temperature change at a given d-value that reduces the population by 1 log value or log unit is known as the z-value. So now we are looking at some of the ways that the mechanical removal methods rely on the barriers. So let's take a look at some of the processes by which we remove things. So we will talk about the filtration process here. So the filtration reduces the microbial population or sterilizes the solution of heat-sensitive material by removing microorganisms. Remember that it was only written that filtration only sterilizes, but here you can see that it has been corrected, that it reduces the microbial population. And we also reduce the microbial populations in the air that we just discussed at the very first slide. So here are some examples of these membrane filters. So to reduce the number of microorganisms, what we used earlier used to call as a depth filter. So this depth filter used to have these channels through the material in a very zigzag fashion, and the microbes used to just adhere onto the material while traveling through those channels. That would reduce the number of microbes. On the other hand, nowadays what is used are these porous membranes with defined pore sizes that remove microorganisms by physical screening process. And here, these membrane filters, they do have a pore size of 0.2 micron. So that is essential that through the 0.2 micron, most of the bacteria will not be able to pass through this and it will be trapped. These membrane filters can be made up of cellulose acetate, nitrate, polycarbonate, then polyvinyl. There are many different types of membranes that is available. The problem of that as their pore size is very small, if you try to filter a liquid through them, it is very difficult for the liquid to pass through. So what you have to do, that here is a setup where you have a membrane filter attached onto this bottle top or a flask top, and you need to use a vacuum pump to suck the liquid in through this material. Also, you can use a pressure to do this. So let's take a look that here is another housing where in this housing right here, you have these membrane filters. And usually most of the time that is used, that this can be used on a bottle top. This can also be even used as a syringe filter. You can actually attach this filter into the syringe and push the syringe, and the pressurized liquid and sterilized liquid is going to come out from the other end. So these are delivered in a sterile package. You open the package and it's disposable. It is one-time use only. And you can easily see that this is a cross-section of a schematic of some of these membrane filters where all the bacteria are trapped at the top. And again, as I said, these 0.2 micron filters, although can remove all different kinds of bacteria except one type, which we called as a mycoplasma, and they cannot keep the viruses out of these liquid as well. So it is sterilization in all different senses except virus and mycoplasma. Here we are looking at the mechanical removal of the particulate air, and this is a high efficiency particulate air filter, or the HEPA filters are used. So there are many different kinds. A surgical mask is also filtering the air as well, but it is essentially disinfection or reduction of the microorganism, but not sterilization. So here what you are seeing, that one of these technicians is working what we call as a biosafety cabinet, also known as a laminar airflow hood. So what is the design for this process? That you have a chamber, and in front of this chamber, you do have a glass sash. You can just completely close the chamber, or you can lift it just a little bit, and you can just put your hands, gloved hand inside, and you can work in this chamber. So how this does work? In the schematic, what you can see, the environmental air is sucked in, and through a blower, it is circulated through a HEPA filter, and the HEPA filtered air is pushed into the chamber, which is in that. And this creates a positive pressure into this air cabinet. So when you see that there is a laminar, why we call it a laminar airflow hood? Because this HEPA filtered air is putting a positive pressure, so nothing from the environment can come inside, even through the opening, because there is a positive pressure inside. And it is also recirculated, and extra air can go outside into the environment through a HEPA filter as well. So this is the working of a laminar airflow hood, where you can use some of the pathogens behind this airflow hood, and it keeps the personnel who is handling the microorganism completely safe, and also keep the laboratory or the environment safe, so that these microorganisms cannot come out of these laminar airflow hoods. But there are some kind of microorganisms which are extremely deadly microorganisms. Say, for an example, we talked about this Ebola virus, Marburg virus, Lassa fever virus. You cannot use an open hood like this. You do have some gloved insertion in there, and you have to use a gloved hand to a completely enclosed biosafety cabinet. But that is something different. So here you can see that different types of filters can utilize to reduce the number of microorganisms in a particular area. Then we are looking at the physical control method, and we are going to talk about the heat and the radiation control. So the moist heat is very effective, and it can destroy viruses, fungi, bacteria. Boiling will, on the other hand, not destroy endospore and does not sterilize, but it will disinfect. The way the microorganisms are killed by the destruction of the nucleic acid or denaturation of the nucleic acid denatures the protein and disrupt the membrane. And the proteins are easily coagulated just above 60-65 degrees Celsius, and it will denature all the soluble proteins and the enzymes, leading to death. So here we are looking at some of the different entities and how they are sterilized or disinfected. So yeast, the vegetative cell, 5-minute exposure to 50-60 degrees will kill the yeast cells. On the other hand, the spores need about 70 to 80 degrees Celsius, about 5 minutes. Molds, 30 minutes at 62 degrees, 30 minutes at 80 degrees Celsius. Whereas for the mesophilic bacteria, the ones that grow at body temperature or around 40 degrees Celsius, 10 minutes at 60 to 70 degrees Celsius to kill all the vegetative cells. So boiling is great for these. But see that 2 to over 800 minutes at 100 degrees Celsius. So this is very important. In the boiling, you can easily understand that they can survive forever in the boiling water. On the other hand, you can see that they are just only viable about half a minute to about 12 minutes at 121 degrees centigrade. So how do you achieve that 121 degrees centigrade? You can achieve that temperature only under pressurized condition, which we are going to see in the next slide. So on the other hand, the viruses can be killed in 30 minutes at 60 degrees Celsius. On the other hand, the prions, 90 minutes at 134 degrees Celsius can kill these prions. So if you read the nice story about the chapter opener, what was said that particularly a person went for a neurosurgical procedure in the hospital and then contacted these prion protein disease and due to die due to that. Remember that the prion proteins are almost very difficult to be sterilized by, see, the sterilization temperature is around 121 degrees Celsius for most of the things. And most of the other sterilization methods will not destroy the prion proteins. Mortality rate for the prion is 100%. If a person gets that, eventually at one point of time, that person will die. There is no cure. So this is a scary thing, that regular processes cannot destroy the prions. So let's take a look at how we do this steam sterilization. So what you're seeing that is carried out above 100 degrees Celsius, which requires saturated steam under pressure. And this process is known as autoclaving, and the machine is an autoclave. So here you can see an autoclave and a cross section of the schematic. Think about that this is a very large, sophisticated, automated pressure cooker, basically. And this is effective against all types of microorganisms, including the endospores. And there are many different ways that we can quality control that. So how this process essentially works, that you do have a jacket which is constantly charged with the steam, and you just put your material inside this chamber, you close the door, there is a silicone gasket in there, and you close the door and you turn on the process, and first it will purge all the air, and then it will increase the temperature, basically sterilize. There are different cycles that go on. If you do have some of the material which are completely dry, doesn't have any liquid, you can do a process which usually we called as a gravity type of autoclaving, where the material is completely dry. It is steam sterilization, but the process is a little bit different. Initially the air is sucked out by vacuuming, and then the saturated steam fills up the chamber. Whereas there is another process by which we sterilize the liquids in that process. You cannot apply that vacuum initially because the liquid will boil over. So essentially the air is removed by purging steam, and the valve is open until all the air is out and the chamber is saturated with the steam. Why we need to purge all the air out? Because what happens that if there is remaining air, it will not achieve the 121 degrees Celsius under the pressure, 15 pounds of pressure. So that's one of the reasons that we need, for the proper sterilization, we need to purge out all the air, and it has to be all saturated steam. So there is also another thing that goes with that how do we know that the material has been properly sterilized? So there are some specific things that has been used. There are some autoclave tapes, and they have some white marks on them, and you can put them onto the material that you are sterilizing. And after they come out, you will see those white stripes have turned into black stripes. So those are heat sensitive stripes. What we mean by that if the temperature of the chamber reaches 121 degrees Celsius, then those white stripes will turn into the black stripes. But still, there could be some problems with that. It is not the best process. If there is a very sensitive process that you need to know that whether the sterilization standard is maintained or not, then you can buy some of these ampules which do contain a bacteria which is known as the Geobacillus stereothermophilus. So the Geobacillus stereothermophilus ampule is put into this chamber, and it is put through the sterilization cycle. Once the sterilization cycle goes on, it will reach that 121 degrees Celsius temperature, and then the ampule is broken and the Geobacillus is put into a culture. If the bacteria fail to grow in the culture, then you know the standard of the autoclave is great. So there is a little bit of modification of this process, which is not sterilization, but used in many different purposes, which is known as tyndallization. So this tyndallization is used for some of the heat sensitive material, and the tyndallization was discovered by the British scientist physicist John Tyndall. This tyndallization process is used in commercial pickling process, commercial canned vegetables, particularly for the tomatoes. And the process uses a steam for about 30 to 60 minutes to destroy the vegetative bacteria. And this process of steaming is repeated about 3 times. And in between these steaming process of 30 to 60 minutes, there is an incubation of this material for about 24 hours. So the purpose of this process is that after the first steam treatment, most of the vegetative cells or all the vegetative cells are dead. There may be some remaining endospores. You give them 24 hours time to germinate into vegetative cells. Then you do the second round steam exposure that will kill all the vegetative cells. Then you do the same thing. So the purpose is basically allowing all the endospores to germinate and killing them in the vegetative cell state, and that is achieved by reducing or destroying almost all the endospores naturally, because there is no endospores left in that material. This is also another process that can be used called tyndallization. So the next one is a pasteurization, and this is controlled heating and temperatures well below boiling temperature. These are used for milk, beer, and other beverages where boiling will spoil them. So Pasteur first initiated that process where he heated the material, particularly wine, at about 55 to 60 degrees Celsius to preserve. Now here, process does not sterilize and does kill pathogens present and slow the spoilage by reducing the total load of organisms present. So you can easily see that when you purchase milk, there is a label on the milk that by which date it has to be used. So beyond that, it should be thrown out or it will get basically spoiled. So you easily can understand that that is only possible due to pasteurization. If you have unpasteurized milk, it will spoil much faster compared to that. So this increases the shelf life of this milk, beer, and other beverages. So there are two processes that is used by the pasteurization. I'm just adding that is not in detail in your book. One method is known as a batch method. In the batch method, a large amount of these beverages are heated at 63 degrees Celsius and it is held at that temperature. In the batch method, a large volume of these liquids are heated at 63 degrees Celsius or 145 degrees Fahrenheit and kept at that temperature for 30 minutes. So it's a 30-minute hold at 63 degrees Celsius or 145 degrees Fahrenheit. So that is one process of batch pasteurization. There is another process of pasteurization which is known as a flash pasteurization, where the pasteurization liquid is passed through a heat exchanger and very rapidly it regains the heat and again cool down. So you have a cooled liquid. Say for an example, for milk, this is routinely used. You have milk at 4 degrees Celsius. It passes through a small heat exchanger material where it is very quickly brought to a temperature of 72 degrees Celsius, and the exposure time is just only 15 seconds. And just after 15 seconds of exposure, it is immediately brought down to 4 degrees Celsius again. So this is known as the flash pasteurization, routinely used for milk pasteurization. So on the other hand, the dry heat sterilization is much more less effective. So for an example, if we just give an example, Clostridium botulinum, you know that it is one of the causal microorganisms for food poisoning. Botulinum toxin is very, very toxic. So the endospore, this is endospore forming Clostridium botulinum, and this Clostridium botulinum endospores can survive about just only 5 minutes at the moist heat at 121 degrees Celsius, which is autoclaving. In autoclave, 5 minutes, all the spores of this microorganism is dead. On the other hand, if you have 160 degrees Celsius dry heat, these spores can survive for more than 2 hours. So you can easily understand that dry heat is much less effective. So most of the times, the items are subjected to 160 to 170 degrees Celsius for 2 to 3 hours. Of course, you cannot do dry heat sterilization of any liquid as such. So that's the drawback. And the process is that it oxidizes the cell constituents and denatures the protein by breaking the hydrogen bonds. And the advantage of that when you do autoclave, no matter what, the steam tends to corrode the glassware and metal instrument as moist heat. But in the dry heat, that doesn't happen. This is the only advantage of dry heat or oven sterilization. So here also what we do, that this is a benchtop incinerators are used to sterilize inoculating loops used in microbiology laboratories. Inoculating loop are basically nichrome wires, which does contain a small tiny loop, which is about 2 to 5 millimeter in diameter at the very tip of the loop, which is connected to a handle. So what you do that you incinerate that before transferring different bacteria. You may have many different species of bacteria or genus of bacteria that you are handling in the laboratory, and you will be using the same inoculating loop, but each time you can use this. This is called a micro incinerator, where you have a ceramic coil inside right here, and that is glowing hot, and you just put your inoculating loop inside that, and it will be incinerated, and in 5 to 7 seconds it will be completely sterilized. You just briefly wait to cool it down a little bit, and then you can use that to transfer your microorganism. Now, routinely before these sophisticated electrical incinerators, a typical Bunsen burner or a spirit lamp was routinely used to burn or incinerate these loop before performing a microbiological procedure. So this is very, very common. So the next one, the second physical method. So we talked about the first physical method, which is the heat, and we talked about the moist heat and the dry heat. Now we are looking at the radiation. First, we are looking at the ultraviolet radiation, and the wavelength is 260 is the most effective or bactericidal because the DNA absorbs at this wavelength. And also, this is a non-ionizing radiation. So what it caused that this ultraviolet caused the thymine dimers formation. We have talked about these, that in between the A and T, AT bonds, it breaks those two hydrogen bonds. And if anywhere in the DNA, there are two T's next to each other, you have a T and another T, the corresponding A and T bonds are broken, and a dimer is formed by a bond in between the two adjacent T's. And here it prevents the replication and transcription, although there are some bacteria which can repair that, but many of these bacteria can die due to the cause of this thymine dimer formation. And the ultraviolet is limited to surface sterilization because it does not penetrate the glass, dirt films, water, or other substances as well. So these are routinely used to sterilize, say for an example, biosafety cabinets or laminar flow cabinets that we just saw. Say for an example, right here. So in the front, there is acrylic or glass, which is not UV penetrant, and you just lower down that glass shield in there to completely close that biosafety cabinet, turn off the light and turn on, there is an UV light inside that, and you can turn it on for about 30 minutes to 45 minutes, and the surface will be sterilized after that. So this is how it is used. Routinely used also in operating chamber or operation theater as well. So on the other hand, this is also used for water treatment, and these can be even installed in the household or small restaurants where you do have a chamber where you do have this powerful UV light, which is emitting 260 nanometer wavelength. Next to the UV light, you do have a clear water chamber or water pipe through which water is flowing, and this is usually made with the UV penetrant glass or plastic through which the UV can pass into the water. So a thin stream of water is passed through that, and as the water is passing through that thin stream of water, the ultraviolet can sterilize or kill the microorganisms that are present in that portion. So these are some of the applications of this ultraviolet radiation. The next one is the ionizing radiation. So here what we are looking at, that you are looking at an ionizing radiation room right here, and the materials are used with a conveyor belt that go into the sterilization chamber. Usually the radioactive source is underneath through which the radiation comes out to sterilize that. So the main reason that what are used, this is also known as the cold sterilization. So this is used for the pasteurization of antibiotics, hormones, sutures, plastic disposable supplies like plastic Petri plates, because they are made up of polystyrene and any autoclaving or dry heat will destroy them. And also food as well. They destroy bacterial endospore, but they are just only, the drawback is that not always effective against the viruses. There are many different types of radiation. So the first one is the gamma radiation. So the gamma radiation penetrates deep into objects. So the sources of the gamma radiation are usually a cobalt-60 source, or it can be a beta radiation, which is accelerated electrons from a high voltage electricity. The gamma radiation penetrates deep into the object, whereas the beta radiations are used for mainly surface sterilization or say plastic wares where they do not have to penetrate deep into the material. Say for an example, in the food industry, the meat products are routinely irradiated, and this reduces the number of this rogue E. coli O15787, reduces Staphylococcus aureus, or also there is another bacteria called Campylobacter jejuni. So this Campylobacter jejuni is also a gastrointestinal disorder causing bacteria. These are all reduced by the radiation on the food products. So the next, what we are looking at, that how the microorganisms are controlled with the chemical agents, and there is a good number of that, and we're going to talk about each one of them. So the first, again, just to remind you that disinfection is ideally effective against a wide variety of infectious agent at low concentration in presence of organic matter. That's the requirement. On the antisepsis is an overuse of antiseptics such as triclosan has selected for triclosan resistant bacteria. This is a problem. So for an example, this triclosan is many times are present in all these antiseptic material. And when you have that, and they are present in even mouthwashes, and sometimes there could be triclosan resistant bacteria that is growing into the tooth plaque, and that is also a possibility. And there are some chemicals which can also sterilize. We're going to talk about that as well. So first, let's take a look at the first compound, which is known as the phenolics. So the phenolics will have a phenol ring, which is just a benzene ring with a hydroxyl, or you can have an orthocresol with a methyl group right here, or you can have an hexachlorophen where two benzene rings connected by a CH2. So this was first discovered by Joseph Lister. He didn't discover the phenol, but he first identified that phenol is a great disinfecting agent. And still today, that the mouthwash Listerine is named after Joseph Lister. So these are commonly used in the laboratory and the hospital disinfectant, and they act by denaturing protein and disrupting the cell membranes. That's their target. So when you are studying about all these materials, just make sure that most of the times the questions that are asked, that where is the target, that's also very important, and what are the specificities. And it is also tuberculocidal, that effective in the presence of organic matter, and pretty long lasting. So one of the disadvantages that, and again it's arguable, is disagreeable odor and can cause skin irritation at a higher amount. But again, it depends. So for an example, any of these house cleaning agent, which we commonly say as a Pine Sol or Lysol, when you have this original Pine Sol-like smell, that is the smell of phenolics. And there could be addition of other odorant to that, but those are all phenol-based disinfectants that we use in the household, hospital settings, everywhere, very effective. The next is the alcohols, and mainly two types of alcohols are used for the disinfection purposes. They cannot sterilize. The same way the phenols can sterilize either. So one is the simple ethanol, and the other one is isopropanol, and widely used as a disinfectant and antiseptics and sanitizers. Commonly used, these are the two. They can be bactericidal, fungicidal, but not sporicidal. They can inactivate some of the viruses as well, particularly the viruses which are enveloped, having a membrane, are completely killed by the alcohols, whereas the ones which are naked viruses, just have a protein capsid, doesn't have an envelope, are resistant to these. The process by which they act is that they denature proteins and also possibly dissolve some of the membrane lipids. So usually what happens, that 70% alcohol is more effective than much more concentrated, like a 95% or 100% alcohol. Most of these ethanols or the isopropanols are used in a concentration of about 60 to 80%. The presence of water along with the alcohol is very important for killing of the microorganisms. Can be routinely used on the skin surface, and just before a shot, putting a needle on the skin, very, very commonly used. The next one is the halogens, and the halogens, there are many different types. One we are starting with is iodine. So when we use the iodine, we use what is called a tincture of iodine. The tincture is basically a 2% of iodine and in a water and ethanol solution with potassium iodide. Now, what they do that they are skin antiseptic, oxidizes the cell constituent and iodinates protein. When they iodinate the protein, they will denature. So at high concentration, they can also kill the endospores as well. So say for an example, what we just can find like a betadine, your over-the-counter iodine, these can cause a little bit of skin damage, staining, or sometimes allergy is a problem of these typical tinctures. So in case of that, you can use what is called an iodophore. So iodophore is basically iodine, which is complexed with the organic carrier. They are water soluble, they are stable, and they are non-staining, and they release the iodine slowly. So as they release the iodine slowly, it minimizes the skin burns. But these are very popular antiseptic agent as well. So what you are seeing on the other hand, right here, before going into the next slide, that this is a compound known as a halazone. And we're going to talk about that. These halozones is a halogenated compound which releases this chlorine in a much slower way. And we're going to talk about that campers and hikers use these tablets to purify the water. And we're going to talk about that. We'll come back to this. So the next halogen is chlorine, and it oxidizes all the cell constituents. And it may be applied as chlorine gas like Cl2s or sodium hypochlorite, NaOCl, which is the sodium hypochlorite is your common bleach, or it could be in the form of calcium hypochlorite. So all of them actually do produce hypochlorous acid, which is HOCl, upon reacting with the water. So almost all microorganisms are killed within the 30 minutes, but there are two eukaryotic pathogens, and both of them, unfortunately, are waterborne pathogen. One is Cryptosporidium and another is Giardia. So the Cryptosporidium cause gastrointestinal disorder. Giardia also do the same, known as giardiasis. These are not killed by chlorine. So that's a problem. And as I said, the halazone, on the other hand, is a water purifier tablet that releases that chlorine to disinfect water in about 30 minutes. So it takes a little bit time, but it also can decontaminate the water. And mainly, these are used, as I said, by the campers and the hikers if they do not have a water supply which is safe. So environmental water from a river, from a lake can be utilized, that we have seen in the last slide. So here, what you can see that the swimming pools, dairy food industries, effective in the household detergents, we use this chlorine or bleach all the time, even in laundries as well. So they can destroy the vegetative bacteria and the fungi. On the other hand, the chlorine gas we do not utilize in the household, but it is sporicidal as well. So these are very powerful disinfectant, as well as if you use the chlorine gas, it could be a sterilant as well. So the next one is a heavy metal, and these are the ions of mercury, silver, arsenic, zinc, copper. And although historically many of them were used, the usage of these heavy metals are coming down. Although historically many of these heavy metals were routinely used, they're effective but usually toxic. And that's the reason that we do not use them anymore. But you can still see that physical presence of that. Think about that, that your hot water pipe are usually made up of copper. And this copper is anti-algal. So the algae, due to the hot water passing through those, will not grow inside those pipes. And that's the reason it is not on the cold water pipe, but the hot water pipe uses this copper to reduce these blooms of these algae which can grow in the hot water pipe. And they basically combine and inactivate proteins and also precipitate cell proteins. But there are some heavy metals that are still used. Say, for an example, your silver sulfadiazine is used on the burns. On the other hand, say, for an example, copper sulfates are used in swimming pools. Usually what they do that they combine with the sulfhydryl SH group of the amino acid, say cysteine, and they basically denature them. That's their mode of action. So the next one, what we are looking at, is the quaternary ammonium compounds. And these are detergents that have a broad spectrum of antimicrobial activity, and they are effective disinfectants. So these are also known as amphipathic organic cleansing agents. And here you can see that there are two examples of cetylpyridinium and benzalkonium. So what they are, that they do have they are amphipathic molecules. So as they are amphipathic molecules, they do have a hydrophilic end and they do have a hydrophobic end. So the hydrophilic end is with an ammonium ion and it is in the quaternary state, and the nitrogen do have a positive charge. Right here, this is the quaternary nitrogen, 4 bonds, and this have a positive charge. So this is the charged hydrophilic end. And as it is a positive charge, you can easily say that it is a cationic detergent. On the other hand, you can see that this C6H32, which is a long chain of hydrocarbon, that is the hydrophobic end. And here, this benzene ring with the chloride, this is the hydrophobic end, as well as right here, CnH2n, which could be many different numbers. And this is also your hydrophobic end. So what basically do that, they kill most of the bacteria, but they are not effective against mycobacterial tuberculosis or endospores. They are stable, non-toxic, and although they are inactivated by hard water and the soap. So how do they exactly work? So the working of these is basically, think about this, that the hydrophobic portion readily goes to the plasma membrane. As they are amphipathic, they do have this hydrophobic end, and they disrupt the membranes. Often they make holes into the membrane and the cellular material will get exposed. On the other hand, think about that, Mycobacterium tuberculosis do have a very special type of material on its cell wall, which is known as a mycolic acid, and this quaternary ammonium compounds of the detergents cannot penetrate that. Endospores are also extremely tough, that cannot be used for that. So most of these sprays that we use as a daily disinfectant, these are your quaternary ammonium compounds that are used routinely in the household sprays to disinfect your kitchen table surfaces or household surfaces. These are also known as the quats because they contain this quaternary ammonium compound, which is right here. That's why they are also popularly known as quats. The next one is an aldehyde, and there are mainly two of them, which is a formaldehyde and a glutaraldehyde. And they are highly reactive molecules. They are sporicidal and can be used as chemical sterilant as well, but they are very irritating to mucous membrane. So you have to use precaution. They do have a strong odor, but they will immediately attack the mucous membrane. So the glutaraldehyde is usually used in 2% buffered condition, and it's a very effective disinfectant as well. On the other hand, formaldehyde is routinely used in the laboratory and the hospital setting as well. These do also have a disagreeable odor. That's one of the drawbacks. So let's take a look at how these actually work, or as a result of the glutaraldehyde. So this is the structure of the glutaraldehyde. So as a result, what happens that the proteins are alkylated and cross-linked to other proteins, which inactivate them. So all these amino groups in the peptidoglycan are alkylated and cross-linked, which prevent them in participating in any other chemical reaction, and those involved in the synthesis of the peptidoglycan. So you can easily see that this is the process by which, right here, amino groups in the peptidoglycan in the gram-positive bacteria, or they can attach onto the protein and they alkylate the protein. And these are the different processes by which these polymerization can lead to the death of the microorganism. So what you are seeing that this is the sterilizing gases used to sterilize heat-sensitive material. These can be microbicidal or sporicidal. Ethylene oxide is carried out in equipment resembling an autoclave, and many times these are used to disinfect like disposable hospital gowns before they are being thrown out. They can be used for those. Also, vaporized hydrogen peroxide can also be used. So let's first take a look at what ethylene oxide does. So what you are seeing that this is a photo of an automated ethylene oxide sterilizer. So here is a chamber where the material that needed to be sterilized are put in there, and then you shut the door, and there are these ethylene oxide and carbon dioxide bottles. These are mixed together, and the material is exposed for a specific time, and then that material and the gases are then removed by an outlet, and then the chamber is open to remove the material. So usually the concentration at which it works, that is a 10 to 20% of ethylene oxide is mixed with carbon dioxide or dichlorofluoromethane. And at 38 degrees Celsius, this process takes about 5 to 8 hours. Whereas if you use 54 degrees Celsius, the chamber temperature, then you can achieve this within about 3 to 4 hours. But you have to maintain a relative humidity of about 40 to 50% in the chamber, and your ethylene oxide concentration should be about 700 milligram per liter. That's what you need to achieve to sterilize completely. On the other hand, the chlorine dioxide gas can also be used as a disinfectant agent. Now, do not confuse that with the chlorine gas. It is very different. That the chlorine dioxide is a very different compound. It directly attacks many of these amino acids and denature proteins, and it can also attack the free fatty acids, which can denature the membrane as well. There are some historic perspective of this chlorine dioxide gas. So we talked about this in somewhere else in 2001 when the attack of the senators happened to this potential bioterrorism attack using the anthrax spores. The Senate chamber as well as the postal facilities were sterilized using this chlorine dioxide gas. This is written in your textbook. You can just read that story as well. So on the other hand, the biological safety cabinets that we saw several pictures of, they can be decontaminated by using vaporized hydrogen peroxide right here. So these are used at a concentration, wide variety of concentration in between about 140 to 1400 parts per million is the concentration, and that can also disinfect or decontaminate, but not for sterilization. And it is pretty safe to use in the sense because the hydrogen peroxide is broken down into oxygen and non-toxic water. So it is pretty safe to use. So these are some of the functions of these sterilizing gases. Ethylene oxide can sterilize. Also, high concentration of chlorine dioxide can do that. On the other hand, vaporized hydrogen peroxide can basically decontaminate or disinfect. So next, what we are looking at that how these antimicrobial agents can be evaluated for their effectiveness. So the evaluation is a complex process which is regulated by the US federal agencies. One is an EPA, which is an Environmental Protection Agency that regulates the disinfectants. On the other hand, the FDA or the Food and Drug Administration regulate the agents used on humans and animals. So the next chapter, which is the antibiotics, almost everything which is being used on the human beings are regulated by that. On the other hand, there are some of the material that we talked about here, particularly the antiseptics, those are under the FDA's as well. So we're going to talk about some of these things. So what are the conditions that influence the effectiveness of antimicrobial activity? So the first thing that is important is the population size of the microorganism. Larger the population, it takes longer to kill than the smaller population. So this is very important. The next important thing is the population composition, where the microorganisms differ markedly in their sensitivity to antimicrobial agents, and also the concentration or intensity of an antimicrobial agents. Usually the higher concentration kill more rapidly, but the relationship is not linear. Say for an example, if you increase the heat, usually the killing process goes faster. Acidic pH kills faster than the basic pH. Say for an example, it is much more difficult to pasteurize tomato juice or lemon juice or orange juice compared to the milk because milk is basic, whereas these are acidic. Also, the presence of organic matter. If there is a presence of organic matter, then it takes a much more higher concentration of the disinfectant to kill the microorganisms. So here, the contact time, longer the exposure, more microorganisms are killed. Higher temperature, enhance the chemical activity, kills faster, and local environment that I was talking about. pH, viscosity of the medium, concentration of organic matter, and so on can profoundly affect effectiveness. And the organisms in biofilms are less susceptible to many antimicrobial agents as well. So all of these we just talked about in the last slide. So the next one is that how do you know that you have an effective chemical agent? So to do that, one is what is called as a phenol coefficient test. This looks for the potency of a disinfectant is compared to the phenol. And this is useful for screening, but it could be misleading. So let's take a look at how do you do this phenol coefficient test. So a series of dilution of phenol and the disinfection being tested are prepared. And then the standard amount of a particular bacteria, which are the Salmonella enterica serotyphi and Staph aureus, are added to these dilutions and then placed at either 20 degrees Celsius or 37 degrees Celsius in a water bath. And then every 5-minute interval, the samples are withdrawn, and those samples were taken and inoculated into a growth media. And then this growth media is incubated and examined for growth at least 24 hours. So from that growth, it is taken that the highest dilution means that the lowest concentration of the testing material that kills the bacteria after 10 minutes of exposure, but not 5 minutes. This is important. The highest dilution of the testing concentration of the material which kills the bacteria after 10 minutes of exposure, but not 5 minutes, so it should be in between that, is used to calculate the phenol coefficient. So how you calculate that? The higher the phenol coefficient value, more effective is that particular disinfectant. So if the value is greater than 1, it is actually better than phenol. If the phenol coefficient is 0.5, so it is half effective as phenol. If the phenol coefficient of a disinfectant is 2, that means it's twice as effective as phenol. As I have said before, that is usually good for screening, but this could be a little bit misleading. So what is more important, that the use of a dilution test which determines the rate at which the selected bacteria are destroyed by various chemical agents. So how this is done, there are some stainless steel carriers are contaminated with specific bacteria in controlled conditions. And these carriers are then dried briefly, then they're immersed into a test disinfectant for 10 minutes. Then they are transferred to a culture media and incubated for about 2 days. And from there, the concentration which kills the bacteria, say 59 out of 60, or say with a 95% confidence, that is basically determined. That is the particular concentration of that particular disinfectant, that at which concentration it should be used with a 95% confidence. So the normal in-use testing is done using conditions that approximate the normal use of disinfectant, and this is the final process that is being used. So the last part that what we are talking about is the biological method. And again, this is not a very mainstream method yet, but we're going to discuss some of the things that what is possible in the future. So again, as I said, this is an emerging field showing great promise. And one of the ways that it can be controlled is by predation by this Delovibrio. So Delovibrio is a predator for Salmonella, for Shigella, for the E. coli, all these gram-negative bacteria. And that can be used. It is not used in the human beings, but what is being used, that they can be sprayed into the poultry, and that can reduce the contamination of these particular meats by the Salmonella, Shigella, or E. coli. What happens, there is an interesting story about the phage, that in the 1900, Felix D'Herelle from the Pasteur Institute in France isolated a bacteriophage from a patient who actually recovered from a bacillary dysentery. And he did a number of experiments, and he came to the conclusion that that particular isolated bacteriophage does have the killing property of that particular bacillus. So it is a bacteria killing viruses. And that was basically the first promise of bacteriophage. So now the bacteriophage itself can be used for some of these control of the pathogens. There are only 3 countries at which it is used on human beings. It is Russia, Poland, and Georgia. No phage therapy on the humans are approved at present in the United States. The US FDA has approved a bacteriophage spray on foods to destroy mainly Listeria, Salmonella, and the E. coli, because the concern for not using in the human beings, because it is kind of a little bit unnerving that if you're just giving live virus to eat to take care of your gastrointestinal disorder by a pathogen. So that's one of the reason. So on the other hand, there is another way that the scientists are thinking about this, that many of these bacteriophage also do produce, which is called the endolysins. These endolysins are basically utilized to lyse these cells or the bacterial cells. So whether these purified proteins can be used as a therapeutic agent, that is also one of the areas of research. Also, another process is that in the environment, many of the bacteria produce these bacteriocins, which are basically for competition and killing other invading microorganisms. And these are basically toxins produced by the microorganisms to kill other microorganisms. And these can be utilized as well. So one of the example of another thing is that there is a bacterial protein called depolymerase, and these depolymerase can be utilized to remove biofilms. So these are some of the biological control of the microorganisms and shows great promise, and probably we will see them in the future much more. So that's pretty much the chapter, which shows the controlling of the microbial growth can be achieved by many different methods, which are some physical, chemical, and understanding what microbes need to grow and thrive is important for the understanding how to kill them and control their growth. And most of the methods are useful, but many of them has the pros and the cons, and each may not be suitable under particular circumstances. And that is true even for the next chapter that we are going to see, that there are many different types for antibiotics, antiviral, antiprotozoal, and each are not suitable for every cases. So we're going to talk about that. So this will be the end of the chapter 8, which is the microbial control of microorganism.